Welcome to the final lecture in this series of DNA and RNA isolation, and we're going to deal specifically in this video with RNA isolation. Our objectives are as follows. We're going to look at the types of RNA in the cells and the relative abundance of each one. We're going to look at the difficulties that exist with working with RNA, why it is so painful and such a hassle. We're going to look at the sources of RNA's contamination. We're going to look at how to sanitize glassware so that we don't have any problem isolating RNAs. And we're going to look at a very abbreviated version of an RNA isolation method, the generic way that we isolate RNA. Types of RNA. There are three categories. The first one, the most abundant, is of course ribosomal RNA. Ribosomal RNA constitute 80 to 85% of the total RNA. This is followed by transfer RNA, small nuclear RNA, microRNA, and other types of very small RNAs, and these constitute 15 to 20%. And then the final one, and probably the most critical, most important for our work, is the messenger RNA, and this category makes up a very small amount of the total RNA of the cell. Why is it so hard to work with RNA? Well, you remember from your basic chemistry that we discussed the structure of DNA or RNA. One of the differences between DNA and RNA is that DNA has deoxyribose sugar, which means it's missing the 2' hydroxyl group, while RNA has the 2' hydroxyl group. Now, you recall that we say that that hydroxyl group can be used to attack the phosphoester bond that holds the backbone of the RNA together. So it begins to cleave itself or degrade itself using that attack of the hydroxyl group on the phosphate bond. And so if you don't remember that, please go back and review that. And so what happens is that you have sequential cleavage of the phosphodiester bond as a result of the nucleophilic attack by the 2' hydroxyl group. And so that's one reason. And the other reason is that the presence of RNases, and these are enzymes that will digest RNA, the presence of RNases almost everywhere makes it very difficult to isolate intact RNA, because these RNases will chew up the RNA. What are the sources of contamination by the RNAs? Well, number one, bare hands. And you will know that when somebody is doing a lot of RNA work and you go and you open a drawer and you touch their tubes and their reagents with bare hands, they will yell at you. There's a reason for that. It's very difficult to inactivate RNAs. Once you contaminate it, you almost have to use a detergent to inactivate that. Contaminated plastic and glasswares are the other source of that. Unclean lab bench is another source, and then endogenous RNases that are released from the cell. And so these are some of the many sources of the contamination by RNases. Well, how do we prevent contamination? Well, number one, you need to dedicate a room that involves no other work other than RNA type work. In other words, if you have a small interior room, you can just clean it up, decontaminate it, put all the reagents that you're going to use for RNA, all the gel boxes, clean them up with DEPC or detergent, and then once everything is cleaned up, don't do anything else in there but RNA type work. This is a dedicated room, and you'd be better off if you just leave it as that. Treat glassware. Another way to get rid of contamination is treat glasswares and plasticwares with a chemical diethyl pyrocarbonate, and this is called DEPC for short. And you make up a 0.1% solution, and you fill up your glassware or your plasticwares, and you kind of let them sit in the 0.1% DEPC solution, and you let them sit overnight, and then you take them and then you rinse them off, and you autoclave them at about 340 degrees for at least half an hour. This is a strong alkylating agent, and of course, if it alkylates the proteins, it will inactivate the proteins. Or one way to get rid of this also is by using commercial inhibitors. And so here I'm going to go through a very generic procedure for RNA isolation, and this is a very generic procedure, and please remember that we can have different adaptations of this procedure. So this begins as follows, and you need to remember that when you're isolating RNA, you must do it very quickly and freeze the sample down immediately. So the first thing is you lyse the cell with a very strong detergent called quinidinium thiocyanate, and then you extract the lysate with phenol, chloroform, and you can put in isoamyl alcohol like we said last time. Remember, phenol is to denature the proteins and lipids and carbohydrates. The chloroform is to make the phenol heavier, because phenol and water densities are almost very similar. And then isoamyl alcohol is an anti-foam agent that you add to it. So, but you make sure that the pH of the phenol chloroform solution is low. And the reason for that is at a high pH, the nucleic acids, both DNA and RNA, will be in the aqueous phase. But when you lower the pH, the pKa for DNA is much higher than the pKa for the RNA, and so the thing that falls out of solution first will be the DNA. And so DNA is going to move from the aqueous phase, the top phase, and move into the inner phase. And so now we have RNA that is in the aqueous phase by itself. And again, the different types of RNA, because we're dealing with total RNA. And then you precipitate with lithium chloride, again that's salt, and ethanol, and we have explained the use of salt and ethanol. And then you store immediately at -80 degrees centigrade in DEPC treated water. Again, anything that is treated with DEPC must be autoclaved, and when you autoclave it, the DEPC is converted to ethanol and carbon dioxide, so you remove it by autoclaving. So that gives you an idea of how difficult it is to work with RNA, and a very generic procedure for RNA isolation.